Right there? Yeah? Okay, so I'm a graduate student at the EPFL BigBrain project in Switzerland. I work mostly modeling rodent cortex. So this is the first time that I interact with human data and human brains. But so the idea was that the motivation of this project was that when I found about the BigBrain data set and I saw that it was like this very high resolution where I could really, like the cortex was this big chunk that was actually a proper model, proper volume, and not just a surface, as it's mentioned in many studies. So I thought, well, I can apply the methods that I have for rodent cortex, but for the human cortex. And of course, also thinking that having an atlas, a volumetric atlas where you can then integrate data, is the first step towards doing full-scale data-driven modeling, like the kind of modeling that we do at the BigBrain. So I'm going to first show things in terms of the rat sensory cortex that I've worked on, and then the move to human. So essentially, the problem is that cortical geometry is complex, right? So it's not well described by just x, y, z coordinates, because it has mainly three features, I think. First, the volume itself, it's not isotropic. There is a layer architecture, and so there is a preferred direction, so to speak. The symmetry is broken inside that volume, let's say. There's also curvature that varies locally, and then in the rodents, the cortex is smooth, but of course in primates there is also folding, which makes it even more complex. Okay, so the idea is how do we work effectively with this structure that has like all these geometric complexities. So the idea is to define a coordinate system within the volume of the cortex that is essentially going to capture these geometrical complexities. So the main idea is to split the spatial information into one axis that will go through the layers. So it's going to be orthogonal to the layers, and it's going to measure cortical depth. And then we will have the other two directions that are going to be orthogonal and that are going to kind of describe the cortical sheet like horizontally, let's say. Okay? And so I propose a method to derive these things. So essentially, the starting point is to have kind of like a depth field defined inside the volume. It's normalized so that we can say like we are at 0 in the pile surface and then at 1 in the white matter surface. And then the way this method works, this is to generate the coordinates that are parallel to the layers, right? So the coordinate that goes along cortical depth, it can be simply like the gradient of this depth field. But to generate the other two coordinates, I apply this method of constructing a volumetric flat map. So the idea is to have starting points. The white dots would be any voxels inside the volume. And then using this depth field and using the gradient of this depth field, we can draw streamlines that go from normalized depth 0 to 1 and that go through each voxel. And then using these streamlines, we can project or like translate every point to the level set at, let's say, normalized depth 1/2. And then what we can do is we define a mesh that approximates this level set so that we can create a mapping from the voxels to points on the mesh. And then we can apply a flattening algorithm to the mesh that will give us something that is flat, so essentially the unit square. Okay? And this defines the M operator, which would be the flat map that moves points in 3D space to points in the 2D surface. Okay? And of course, we can then discretize the mesh to obtain like integer coordinates of whatever resolution is good for our problem. So having this kind of mapping, this is a volume to surface mapping that can, of course, also be inverted in a sense, which allows doing things like this. So this is the same chunk of somatosensory cortex that I showed you before. And the idea is that we have this flat map operation that if we kind of draw things on 2D, we can then lift them back to 3D. Okay? And when we do this, because of the way the coordinate system is defined, we will always recover things that look like columns, okay, where all the layers will be present and will be preserved in that sense, okay? So this has multiple applications that we have used when doing models with transformed sensory cortex. But essentially what I say is that, oh, maybe I can apply this also to the human cortex. So I did this. Essentially, I took the layer annotations available with the BigBrain dataset and also the boundary meshes that are available. And then, well, I went to some processing steps of the data to have it in the input format for the algorithms that I already had. Essentially, like relevant steps here would be downscaling to 100 microns because, you know, working with the full resolution of the BigBrain can be a bit too much, well, depending on the computer as well. I work here with the supercomputer that we have at BlueBrain, but still, like, loading the full image in memory all at once, it's not feasible. So I downscaled, and then I used the layer annotations to generate the depth map by using Gaussian smoothing on each slice and then on the 3D volume, so as to kind of approximate a solution to the heat equation based on like the prior defined by the layer annotations, which are just discrete levels. And then I could run the full flat mapping algorithm that I just showed you. So in the end, the result is that we obtain, for the geometry of the BigBrain, we obtain a z coordinate that will give us cortical depth at any location in the volume, and also, of course, the direction vectors that point towards the pile surface. So here it's visualized just on the outer surface, but remember that this is defined at every voxel inside the volume, okay? Every voxel will have a vector looking towards the pile surface and a value for the depth. Okay, and then we also have the flat map. So essentially, we have this unit square where we have x and y coordinates, and when we draw like x constant lines, that maps back to kind of ribbons that go from like anterior to posterior along the volume. And if we plot like y constant lines, we obtain ribbons that go from medial to lateral in the volume of the cortex. Okay. So we have this transformation, this flat map that allows us to go like from 2D, which is the mesh, to 3D, and also back. So one application of this is, for example, I just took one of the parcellations that are available also in the BigBrain FTP server. These are like these parcellations that have been mapped, I think, from the MRI surfaces to the BigBrain surfaces, and then I'm mapping from the BigBrain surfaces to the volume using the flat map. And so essentially this transforms an annotation that is defined on the mesh to an annotation that is defined in volume. So like every voxel gets the membership to the region. And of course, this lifting is done like following all these streamlines, so that the direction in which this happens is like orthogonal to the layers and following this kind of columnar organization. So this is one example. This is another of the atlases. So this can be done for like any annotation that you define on the mesh. You can then lift to the 3D space very easily. And then, of course, you can also go the other direction from 3D to 2D so that you can visualize data, for example. So this is two things. Now, on the left, we have cortical thickness. So this is computed at every voxel. And then, of course, when we project to the 2D surface, we have to take the mean or the maximum or some aggregation function. And then we can visualize, in this sense, what is happening in the whole cortex in this kind of unit square representation. Okay? And then on the right, we have something called conicity, which simply means whether when you go deep in the cortex, if it gets narrower or it gets wider, which would be kind of a surrogate for sulci and gyri. And in this way, we can see the cortical folding as well represented on the flat map. So there's like these two applications of having the flat maps, is you can visualize data from 3D to 2D, or you can lift annotations from 2D to 3D. Now the next thing that we want to try is, well, now that we have this atlas geometry, we can kind of start doing the BlueBrain pipeline for building circuits. So I just extracted a chunk of the temporal cortex, and because we have data on temporal cortex, we have electrophysiology, cell densities, etc. So we're going to try and build a model, like a biophysically detailed model of this piece of temporal cortex using this atlas geometry as a basis. Okay, this is still a work in progress. And then finally, some challenges. Of course, one thing is that the atlas geometry is in the end defined by the layer annotations and not the original image, which they differ a little bit now because of the way the layer annotations were defined. And then, of course, this was done at a downscaled resolution. Doing it at full resolution would require a bit of tweaking the algorithms because you cannot load all the datasets in memory at the same time, so some kind of approach would be needed there. And then also to define the gradients, it's important to have like hemisphere masks, because in the point where the hemispheres meet, the gradient is not well defined. So here I had to like manually annotate the 100 micron volume, but of course manually annotating the full resolution thing, it's too much work. So this, if this was provided with the BigBrain dataset, would be very good. And also the layer boundary meshes, because they are made to have this kind of mapping from MRI. They have, for the purposes of this, like too low resolution, so it's just 100,000 of vertices. But here we would like to have like millions of vertices, so that the mapping from voxels to nodes in the mesh would be better. So yeah, higher resolution meshes would improve all of this. And then, yeah, well, some acknowledgements. This happens within the context of the BlueBrain project. These are my main collaborators, my co-supervisor, and the lead of human circuits in the lab, and of course the people that make our supercomputer run, because without it, this couldn't be done. And yeah, so finally, just a bit of self-promotion. If you want to look more into this work or related work, you can check out the preprints. Thank you.